Thank you very much, Dr. Molitor, for this kind introduction. Good morning, dear colleagues. My keynote is about a timely and ill-understood subject, pigs, the role of pigs in pandemics. And I would like to try to discriminate between fiction and those things we really know. So I plan to start with a bit of an introduction about influenza in pigs and in people, and then I will guide you to the most important pandemics of the last century, a pandemic scare, and the first real new pandemic since some 40 years. And this should give us better insights in the role of pigs. These days, only a hermit does not know what the H and N of an influenza virus stands for, or what a subtype is. And you will probably know that there are many different influenza virus subtypes. All of them can be found in wild birds, but only a limited number have become established in pigs and in people. I also want to remind you that influenza is an RNA virus with a segmented RNA genome. And the virus uses two tricks to try to escape from the existing antibody response in a population. Antigenic drift is a relatively small change in the hemagglutinin or neuraminidase, the result of an accumulation of mutations, and it can lead to local virus outbreaks or epidemics. And this contrasts with antigenic shift, which is the introduction of a novel hemagglutinin subtype, and because the existing antibodies do not recognize the novel hemagglutinin, this may lead to a pandemic. And one mechanism for such an antigenic shift is genetic reassortment, which is a process of gene swapping. So when two different influenza viruses simultaneously infect the same cell, the gene segments from virus A can mix and match with those of virus B to form a new combination virus with exactly 8 gene segments. But reassortment is a very complicated process, and only in some cases will the reassortment have a novel hemagglutinin. And this is actually what happened during the pandemics of 57 and 68, when the previously circulating human virus got the hemagglutinin from an avian virus. But those human gene segments help the novel virus to be well adapted to humans. Another mechanism for a shift is interspecies transmission. So there is consensus now that all novel influenza viruses come from wild aquatic birds, but fortunately the species barrier is rather strong, cross-species transmission is rather difficult, and each species has its own sort of influenza viruses. And pigs and people are susceptible to the same virus subtypes, and this makes swine flu research even more interesting. Both in pigs and in people, influenza is the typical example of an acute respiratory tract infection. And this graph here shows mean virus titers in various parts of the respiratory tract of pigs at different time points after an experimental intranasal inoculation. And you will see that we find the highest amounts of virus in the lungs and the trachea. And this is also illustrated by these immunofluorescence stainings for the virus, which show large numbers of virus-positive cells in the bronchi. We find slightly lower amounts of virus and fewer positive cells in the nasal mucosa and the nasopharyngeal tissues. And as early as 6 or 7 days after the inoculation, the virus is completely eliminated. The symptoms are typical of an infection of the lower respiratory tract, and there is massive infiltration of the lungs and especially the bronchi with neutrophils. And those neutrophils really obstruct the bronchi, and this causes severe breathing difficulties with tachypnea and a labored abdominal respiration. But in spite of the fact that the virus replicates only in the respiratory tract, there are also prominent systemic signs like fever, lack of appetite, and dullness. And we and others have found that the simultaneous release of a whole series of cytokines, which are inflammatory mediators locally in the lungs, that those cytokines are at least in part responsible for the typical pathology and disease. And in our experiments, there has always been a strong correlation between cytokine levels in the lungs and the extent of disease. But I have to say that those cytokines are also strong stimulators of the specific immune response, so they have very beneficial effects too. The very same cytokines have also been found in respiratory secretions of humans with influenza. And what's more, it seems that a cytokine storm or an overproduction of cytokines is responsible for very severe human influenza infections, like the 1918 flu and like H5N1 in humans. And there are plenty of other similarities between humans and swine. The sites of virus replication, also the very short duration of the infection. The symptoms are rather similar, and in both species there is a low mortality in healthy subjects. And in my opinion, pigs are one of the better animal models to study the pathogenesis of influenza, much better than mice, which are not natural virus hosts and which show a completely different pathology and clinical signs. But the human and swine influenza viruses do show differences in their antigenic and genetic makeup. And another interesting point is that the swine influenza viruses differ in different parts of the world. So you in the US here have different influenza viruses than we in Europe. Many, if not most, of the swine viruses have emerged through genetic reassortment, and we will come back to that later. This is why the pig is considered a mixing vessel for influenza viruses. And swine influenza is one of the most important causes of acute respiratory disease, but in my experience in the European situation, most pigs become infected without ever showing the typical disease, so the large majority of the infections are subclinical. To illustrate my point about the differences between swine and human viruses, I will make an attempt to explain this complicated figure about the evolution of H1N1 viruses in swine and in humans. So, the swine H1N1 viruses in the US and the early human H1N1 viruses both derive from the 1918 pandemic virus. They have exactly the same ancestor. This swine virus is called classical H1N1, but the two viruses have evolved along different pathways, and the result is that the recent swine and the recent human seasonal H1N1 virus are antigenically different. The swine H1N1 virus in the US has also undergone some complications because you had introduction of novel viruses in the meantime, especially H3N2, and those viruses have started to reassort with classical H1N1 to give the virus a novel set of internal genes, a so-called triple reassortant internal gene cassette, but the hemagglutinin remains rather similar. In Europe, we have an entirely different H1N1 virus. Our H1N1 virus comes from wild birds. It's very different from classical H1N1, and we call it avian-like H1N1. Those two viruses, the European swine and the North American swine, have reassorted to form the novel 2009 H1N1 pandemic virus in humans. So right now we have two different viruses in the swine population, avian-like H1N1 in Europe, classical H1N1 in North America, and most of them with the triple reassortant internal gene cassette. And in the human population, we have the seasonal H1N1 viruses that have been present since a long time, and since this year, the novel H1N1 pandemic virus. So this is only for H1N1, but also for the other subtypes in swine, the history is extremely complicated, and I will not go into details about that. It's known for a very long time that swine influenza is a zoonosis, but this has never caused too much concern in the past for several reasons. First of all, the total number of documented human cases remains rather small, and most of these cases were clinically mild, very similar to infections with human seasonal influenza viruses. Most humans had had contact with pigs. There have been a few cases of secondary transmission, but in all of the cases, the virus lacked the critical capacity to spread further in the human population. And this is very important. I have to say, though, that almost all of the existing swine flu genotypes have ever been demonstrated in a human. And there have also been a few fatal cases in humans. So this is all based on virus isolation or virus detection in respiratory samples of humans. But there are also serological studies. And the serological studies suggest that much more humans in contact with pigs have antibodies to swine influenza. But they cannot say how many, because there is some extent of serological cross-reactivity between the swine and the human viruses in the serologic tests that are available for these studies. So the authors are aware of that problem, and they try to solve it by using complex statistical methods, but in the end we don't know the exact incidence of the infection in humans, and personally I'm also rather critical to the interpretation of most of these serological studies. So what you should take from this is that there is a variety of genetically heterogeneous influenza viruses circulating in swine populations worldwide, and many of those viruses have only emerged during the last decades. And the true incidence of zoonotic swine influenza is unknown, but until very recently, all of those swine viruses lacked the critical capacity to spread efficiently between humans. So we will now have a look at the pandemics of the 20th century, and there have been three. The so-called Spanish flu of 1918 was caused by the H1N1 virus that we have just seen. It has remained in the human population for a very long time. It temporarily disappeared in the 1950s, was reintroduced in 1977, but the current seasonal viruses are still, so this Spanish virus is still a far ancestor of the current seasonal viruses. The 1957 pandemic was caused by H2N2, and that virus disappeared around 1968 when it was replaced by H3N2. So right now we have three different influenza A viruses in the human population, H3N2, seasonal H1N1, and the novel 2009 H1N1. The 1918 virus was a real killer virus, but the positive news is that successive pandemics tend to become less and less severe. Of interest to us is that the clinical symptoms of flu appeared simultaneously in swine and in humans in 1918. And in fact, swine flu was described only for the very first time in 1918, while descriptions of influenza in humans go back to the Middle Ages and even before that time. Recently, the team of Dr. Jeffrey Taubenberger started to sequence the whole genome of the 1918 virus using fixed and frozen lung tissue specimens from 1918 victims, and they concluded that the virus was an avian virus and that it jumped to humans and to pigs shortly before the start of the pandemics. But other researchers do not entirely agree with this hypothesis, and recently it was completely disputed by a very recent study. So those researchers argue that the 1918 virus was a reassortant and that the parental viruses have been present in swine or in humans several years before 1918. But of course, there is very limited genetic material of that time available, and it's highly likely that we will never know the exact origin of the 1918 virus, and that we will never know whether the virus went from humans to pigs or the other way around. Both the pandemic viruses of 57 and 68 emerged in Asia, and they were reassortants with the hemagglutinin gene and one or more other genes of a novel avian virus and the other genes of the previously circulating human virus. And as I said, the human virus genes help the novel avian hemagglutinin to get adapted to humans. And it's very clear that reassortment involving a hemagglutinin gene is a very important process in the emergence of pandemics. It has been proposed many times that these reassortants have emerged in swine and that they were transmitted from pigs to people. But what's interesting is that it has never been proven that those reassortants have emerged in pigs. It's a mere hypothesis, actually. And in the textbooks, we can also read that pandemic influenza viruses come from wild birds, and they do not jump directly to humans. They use the pig as an intermediate host in which they adapt to mammals by either mutations or genetic reassortment. And this hypothesis is largely based on research findings that you can read here. Those are findings mainly from the 1980s and before. So at that time, it was shown that pigs are experimentally susceptible to almost any avian influenza virus, while it was believed that humans are not. Various reassortant viruses were also isolated from pigs in the field, and it was known for several years already that swine influenza can transmit to humans. But one often overlooks that the swine viruses usually do not manage to spread further in a human population. Now, one key reason for proposing the pig as a mixing vessel and intermediate host relates to the nature of the influenza virus receptors in the pig. And the binding between a virus and its receptor on the host cell is the very first step for the virus to get into the cell and to start a successful replication cycle. And the known receptors for influenza viruses are sialic acids, and they are bound to a sugar with a linkage that we call alpha-2-3 and alpha-2-6. And the important thing is that avian viruses prefer to bind to the alpha-2-3 linkage, while viruses from humans and swine prefer the alpha-2-6 linkage. And this fits with the finding that the human respiratory tract only has the receptor for its own viruses, while birds only have the receptor for the avian virus. But in 1998, it was found that the porcine trachea expresses both the human and the avian receptor. So all this was in favor of the pig being a unique intermediate host and mixing vessel for avian and human influenza viruses. But these are the single established facts about reassortment and swine. So we know that reassortment occurs extremely frequently in pigs, but the established swine reassortant viruses so far failed to spread further in the human population. And on the other hand, it's very clear that the 1957 and 68 pandemic viruses were reassortants, but there is no proof at all that those reassortants emerged in the pig. And during recent years, there have been two very interesting events that started to challenge some of the old dogmas. And the first was the emergence of a highly pathogenic H5N1 virus in birds in Asia. So the history of H5N1 is also very interesting and complex. The virus has been present for more than 10 years now. It has also spread beyond Asia. And the important thing is that it's the most widespread outbreak of a highly pathogenic avian influenza virus ever. It has become enzootic in large areas of Asia, and we will have to live with it for several more years. So this virus has also crossed the species barrier to humans. And surprisingly, all of those humans became directly infected by contact with infected poultry or their feces. So this kind of challenged the traditional viewpoint that the virus has to go through the pig to infect humans. The virus is also exceptionally virulent for humans, but on the other hand, there have been only some 500 reported human cases. And the most important thing is that the virus still fails to spread from human to human. And this is the major reason why it has not caused a pandemic so far, despite all the predictions about an inevitable bird flu pandemic and all those pandemic preparedness plans. These findings have prompted some researchers to reinvestigate whether the avian virus receptor may be present in the respiratory tract of humans. And these are sections of the nose, the bronchi, and the lung of humans. And the avian receptor is stained in red. You can see it here. The human receptor is stained in green. And the major finding was that the avian receptor is also present in the respiratory tract of humans, but only in the lungs, not in the nose and upper respiratory tracts. So this could be a reason why H5N1 seems to replicate very poorly in the nose of humans, and this may be an explanation for the lack of human-to-human transmission of this avian virus. H5N1 has also infected pigs in nature, but the incidence of H5N1 in swine in Asia has remained very, very low, even in regions with widespread outbreaks in poultry. And experimental studies have also demonstrated that pigs have a rather low susceptibility to H5N1. So in these studies, the infection was almost entirely subclinical. The virus replicated only in the respiratory tract of pigs and to much lower titers than a typical swine influenza viruses, and all of the viruses examined failed to transmit between pigs. Now, one difficulty with such studies is that they have to be performed in biosafety level 3 conditions. And this means that you can use only a low number of pigs and that you can do a rather limited number of examinations. And this is why in my group we work with low pathogenic avian influenza viruses. And those viruses are structurally very similar to the high path viruses, but they are harmless for poultry and for people. And one of my students has done a large comparative pathogenesis study of such a low path avian H5N2 virus and a typical European H1N1 swine influenza virus. And you can see the most important findings here. So we did not find a difference in the replication pattern of both viruses. Both viruses replicated in the entire respiratory tract of pigs, and they replicated better in the lungs than in the trachea, and better in the trachea than in the nose. But the avian virus replicated approximately 100 times less efficiently at all levels of the respiratory tract. And the result of this is that particularly in the nose of the pig, avian influenza virus infected cells are very, very rare. And this could also explain why the amount of virus in nasal swabs is relatively low for the avian virus if we compare it with the swine-adapted H1N1 virus. The next question was whether such avian viruses could be able to transmit between pigs. And to find out, we used this experimental design. So we had groups of 6 pigs, and they were inoculated intranasally with one of 6 low-path avian influenza viruses. And 2 days later, they were brought into contact with 6 influenza virus naive pigs and also 2 naive ferrets in a cage. We used the ferret as a kind of model for humans because the pathogenesis of influenza is very similar in ferrets and in humans, and they are one of the accepted models for human influenza. So the outcome of this large study was very simple. None of the viruses could efficiently transmit to contact pigs or to the ferrets. And this contrasted with swine H1N1 and H3N2 viruses. We did similar experiments with both viruses, and both of them transmitted highly efficiently from the inoculated to the contact pigs and to the contact ferrets. Another student of mine has started to reinvestigate the distribution of the influenza virus receptors in the entire respiratory tract of swine. And this was really new because the old studies only focused on the trachea of swine. And here you can see sections of the nose, the bronchi, and the alveoli, and they are stained for either the human-type receptor and for 2 different subtypes of the avian-type receptor. This one is the most important. And we found the avian receptor in the pig, but it was only present in the bronchi and in the alveoli, and it was almost completely lacking in the nose of the pigs. The human-type receptor, in contrast, was present along the entire respiratory tract. And in fact, this pattern is just almost identical to the pattern that was found in humans. So based on the receptors alone, we have little reason to assume that the pig is more susceptible to an entirely avian virus or that it's a better reassortment vessel. In summary, all the experimental studies demonstrate a serious barrier to infection of pigs with a wholly avian virus. And those studies are in line with the notion that avian viruses have to adapt through mutation or through genetic reassortment to replicate efficiently in the pig and to transmit efficiently between pigs. But unfortunately, we are still completely in the dark about the exact nature of such genetic changes and the chances that they will occur in nature. And this is key to the understanding of how and why pandemics occur. So, one of the most interesting events in the history of influenza has been the emergence of the novel 2009 H1N1 pandemic virus in the spring of this year. And it's almost certain that this virus is a reassortant between two existing swine influenza viruses. The triple reassortant swine viruses from North America have donated 6 genes, including a classical H1 hemagglutinin, and the neuraminidase and matrix genes come from the avian-like H1N1 viruses in Europe. But still, the exact genesis of the virus remains an entire mystery, because this specific gene combination had never been found in pigs at the time of the discovery of the virus in humans. So it's most likely that the reassortment occurred in a pig, but we cannot exclude that it may have occurred in another animal or in humans themselves. But one of the major differences between this virus and the well-known swine influenza viruses is that this virus spreads very readily between humans, and we do not know why it does so. And I also want to stress that there are no indications that swine have played a role in the spread of the virus in the human population. You will probably know that there have been cases of novel H1N1 on swine farms in Canada. It's rather difficult for me to get reliable information about that. Some people in the audience may have better information, but the dominant viewpoint is that those swine farms became infected through infected humans and that the virus was absent in the swine population before it appeared in humans. This phylogenetic tree illustrates the evolution of the seasonal human H1N1 viruses from the start of the 20th century until 2008. And the novel H1N1 virus is here, and you can see that it is way different from the seasonal human H1N1 viruses. There is as much as 25% amino acid divergence. The closest relatives are swine H1N1 viruses that have been isolated from humans, and older human H1N1 viruses, those isolated in 1950 or before, are also rather similar to the novel H1N1 virus because they are closer to that 1918 ancestor. And the practical consequence of all this is that humans over 60 years of age, those that have been in contact with H1N1 human viruses from 1950 or before, those people frequently have cross-reactive antibodies against H1N1 in HI or VN tests. And this may also explain why novel H1N1 has hit hardest in people under 25. And there are a number of other reassuring facts. So far, the virus is not particularly virulent or particularly lethal in the human population. Vaccines are being developed and will soon be available. And most of these novel H1N1 viruses are also sensitive to the currently used antivirals, the neuraminidase inhibitors. So, in the meantime, it has become clear from the field and from several experimental studies that pigs are clearly susceptible to novel H1N1. And the experimental studies basically showed that novel H1N1 in swine behaves almost exactly like the established swine influenza viruses, and that it spreads very readily between swine. And this has increased concern that the virus may become more and more widespread in the swine population, and that swine may start to spread the virus to humans. But one critical question is to what extent immunity to the established and zootic swine influenza viruses could offer some extent of cross-protection against novel H1N1. And I'm not going to talk about the US situation. I will focus on the European H1N1 and H1N2 viruses, and those viruses are actually antigenically very different from your North American viruses, and they are much farther away, much more distinct from novel H1N1. So to get a preliminary sense about potential cross-protection between established swine viruses and novel H1N1, we started with a very simple serological study with well-defined sera from experimental studies. And those sera were from swine that had been vaccinated twice with swine flu vaccines on the European market, or from swine that had been intranasally inoculated with live European influenza viruses. And the sera were then examined in hemagglutination inhibition tests against a novel H1N1 virus and a series of North American swine viruses. To our surprise, 2 of the 4 vaccines examined induced cross-reactive antibody to novel H1N1. And we also found cross-reactive antibody in the pigs that had been subsequently inoculated with H1N1 and 4 weeks later with H1N2. And I have to say that both viruses in themselves show only 75% amino acid homology in their H1 with the novel H1. And the general finding was that the higher the antibody titer to the European virus, the higher the chance for cross-reactivity with novel H1N1. So this is a mere serological study, and we continue then with a challenge study. So this is only a very recent study, and in this study, pigs were first infected intranasally with our European swine H1N1, and 4 weeks later, they were challenged with a North American triple reassortant swine H1N1 virus. Actually, this virus is very closely related antigenically to novel H1N1, and we use it as a kind of substitute for novel H1N1, because at the time of this study, we could not work with a novel virus in our biosafety level 2 facilities. So this graph here compares the nasal excretion of the challenge virus, and you can see the challenge control group. So these pigs shed very high amounts of the North American H1N1 virus. Then we had one control group that was infected with North American H1N1 before and challenged again with the same virus 4 weeks later, and that group remained completely negative, all values below the detection limit. And then we had the principal group, the pigs infected with European H1N1, challenged 4 weeks later with North American H1N1, and they were almost completely protected against nasal excretion of the North American virus. And of the 4 pigs that were euthanized after the challenge, only 1 tested positive in the samples of the respiratory tract and to very low titers. And the important issue now that is not on the slide is that the prior infection with the European virus failed to induce cross-reactive antibodies to the North American H1N1 virus. So we find this very strong cross-protection in the absence of cross-reactive HI antibody. So in summary, serologic cross-reaction with novel H1N1 is relatively common after vaccination with European vaccines or after multiple infections with live European H1 viruses, and we did such combined infections because they are extremely common in the field. We also found cross-protection against infection with a North American H1N1 swine virus after prior infection with the European virus in the absence of cross-reactive antibodies, and this indicates that European pigs may have partial immunity to the novel H1N1 viruses. And personally, I do not believe that swine will become important spreaders of the novel H1N1 or that they will become a risk for humans, at least not in regions where there is a high population immunity against several other swine viruses. It may be very different in Australia, where there are few swine and no immunity against swine influenza. So during the last few minutes, I would like to come back to those myths and facts. And we've learned that it is a myth that pigs are more susceptible to an entirely avian flu virus than humans or other animals, or that they are necessary intermediate hosts. It's also not true that pigs are the single animal species in which genetic reassortment may occur, or that they are the single animal species with both avian and human-type receptors. On the other hand, it is true that genetic reassortment is extremely common in the pig, and I consider pigs as kind of reservoirs for human and avian flu gene collections. And just very recently, we have a documented case of a presumed swine origin influenza virus that launched a pandemic in humans. And this in itself, the fact that we had to wait so very long for this to happen, this illustrates that a virus that replicates efficiently in pigs will not necessarily be successful in humans. So I hope I have not disappointed you, because in fact we are still lacking insights, the critical insights that are needed to correctly address the role of the pig in pandemics. We don't know which viral genetic changes are needed for efficient replication of an influenza virus in swine and transmission between swine, or for efficient transmission to humans. We don't know whether these adaptive changes will happen more likely in swine than in humans, and we are almost completely in the dark about the extent of cross-immunity and cross-protection between established human viruses and viruses of swine origin. Those questions can only be solved by experimental research in swine with a variety of natural and genetically engineered influenza viruses. And personally, I'm not